Sathyabama Institute of Science and Technology.
Good morning, everybody. This is Jesia Persis Preethi, Assistant Professor from the Department of Biotechnology, Sathyabama Institute of Science and Technology. And today we'll be seeing the mechanism of DNA replication in detail. So, DNA replication is the process in which a new DNA is synthesized, and this is a very well orchestrated and beautifully coordinated together by the enzymes and lot of protein molecules. And first and foremost, for the DNA to be copied into the daughter strand, the DNA strands, which is a double stranded DNA, the strands should be separated. And for this, first and foremost, helicase comes into action. Helicase is an enzyme that hydrolyzes the nucleotide bond, that hydrolyzes the hydrogen bonds which are present between the two nucleotides which are present on the either strands. As the helicase opens up the strand, the two exposed strands, they have the tendency of re-annealing. Therefore, to prevent it from resealing, there are single-stranded binding proteins, which are also known as SSBs. So, these single-stranded binding proteins come into action to ensure that there are no, the strands are not joining back again. So, here it is clearly depicted where the two strands are kept apart and single-stranded binding proteins are ensuring that they are not sealing back again, and this gives room for the DNA primase to synthesize the RNA primers. So the initiation part, it has the main role where DNA primase synthesize the primer, and this primer make sure that there are new DNA. This primer acts as a starter for the DNA to be synthesized. And in this context, we should also speak about topoisomerase. Topoisomerase is an enzyme which helps make nicks, and it also ensures there's no stress developed on the DNA strands. So, this is a classic picture of a DNA polymerase. So, DNA polymerase generally looks like a clamp which has a three domain arms, and this clamp like structure, it's generally uh, attached to the nuclear membrane. So it acts like a clamp which holds the DNA strands apart, as well as it also makes sure that DNA is synthesized in a leading strand as well as lagging strand, both are synthesized. In the elongation context, first and foremost, once the primer is added by the DNA primase enzyme, the synthesis happens in the direction of 3' to 5' on the parent strand, which means that the daughter strand is synthesized from 5' to 3' end. So hydroxyl end which is present, the OH bond which is present is used to create phosphodiester bonds between the two nucleotides. So for this process, it always happens between 3' to 5' on the parent strand. But we know that the DNA strands are anti-parallel, which means that there is one strand which is 3' to 5', whereas the other strand is anti-parallel, meaning that this is 5' to 3'. So, as I said, the synthesis, the DNA polymerase synthesis is carried out from 3' to 5' end. In this context, on one strand, the DNA polymerase smoothly synthesizes the newly forming DNA strand. Whereas on the other strand, as we see, say let us say there is this primer which is synthesized and the polymerase moves in this direction. So this is synthesized. But the problem arises when the DNA helicase, when it unwinds the DNA, the newly opening up strand has to be synthesized, which means that there is requirement of another new primer which has to be employed here. And when the another primer is formed, what happens is another DNA polymerase comes into action, making sure that the strand is synthesized. So in this context, on the strand, anti-parallel strand, what happens is the newly synthesized daughter DNA is synthesized in shorter fragments, which is discontinuous mode. So therefore, it is called as lagging strand. The continuous strand is called as leading strand. Leading strand and lagging strand. The strands which are formed shorter segments of DNA which is newly synthesized on the lagging strand is known as Okazaki fragments. Once the DNA is synthesized completely and elongated, now once it reaches the end of the DNA strands, or once it reaches the next DNA replication fork, the termination comes into action. So the replication of DNA comes to an end with the termination. In this termination, first and foremost, there are important things that has to be done, that is the RNA primer should be removed, as well as the discontinuous joints which are present on the lagging strand should be resealed, which means we have the requirement of other strands. So ligase takes care of this. So ligase, as it moves, it removes the primers as well as it also seals back the discontinuous strands. So this is how the DNA replication takes place, and at the end of the synthesis we have a two newly formed DNA molecules which has one strand from the parent strand and another strand from the newly synthesized daughter strand. Therefore, it is known as semi-conservative process, and this is how the DNA replication takes place. Thank you.